It's my great pleasure, with some ado, to welcome our speaker today, Dr. Maryam Goudarzi. Dr. Goudarzi has an illustrious career and a long one. She did her bachelor, her master's, and her PhD at George Mason University in Fairfax, Virginia, with a 2005 master's in chemistry and a PhD in 2011 in biochemistry with Timothy Bourne on proteomics of extremophiles and their specific enzymes. At the same time, in the Center of Applied Medicine, she did work on cardiac proteomics. And afterwards, from 2011 to 2016, she worked as a scientist and postdoc at Georgetown University in Washington on space and radiation metabolomics from 2011 to 2016. In 2017, she went to Cleveland Clinic as project scientist, and there she worked in the Metabolomics core as team leader for the Untargeted LCMS applications. In 2022, she joined SciEx as senior manager in Metabolomics until last week. And next week, she will start as a senior manager at Charles River in Cleveland for metabolomics and lipidomics. Charles River is well known for its mouse models, but it's also a CRO doing contract research in metabolomics and lipidomics. So today she will tell us her stories about a single human relevant fast food meal and how it produces rapid reorganization of the gut microbiome and host circadian rhythms. Maryam, thank you so much for joining us in person. I think first external speaker for like 3 years, and look what the mess we have. Thank you.
Well, thank you, Oliver, for inviting me and the kind introduction. And to everyone online, thank you for staying with us while we tried to work through the technical difficulties here. So I will talk about one of the studies that I had the fortune of participating in at the Cleveland Clinic on fast food diet and how it impacts the gut microbiome and as a result produces a physiological response in the host. I will give a little bit of background and go over some of the key publications that came out of Cleveland Clinic, especially Stan Hazen's lab and Mark Brown's lab, that really laid the foundation for the study that I'm going to present. Metabolic syndrome, according to CDC now, 1/3 of the adults in the United States suffer from metabolic syndrome, which is a group of cardiometabolic risk factors such as insulin resistance and dyslipidemia. And it looks like that, according to this graph that you see on the left-hand side, the prevalence of metabolic syndrome in adults from 1988 to 1994, and then the next time that it was measured, 2007 to 2012, went up by 35%, which is a significant jump. And there are lots of factors that contribute to metabolic syndrome. And I think, according to CDC, some of the top ones that impact people experiencing metabolic syndrome is education level, socioeconomic status, and advanced age. But we also know that diet can impact and prevalence of metabolic syndrome, especially having access to a lot of fast food, cheap diet. If I show you the map of the United States and the self-reported BMI of greater than 30 in adults in the United States, according to CDC, if you map that out, you can overlay it with, or it trends pretty well with the concentration or density of the food fast food restaurants in the United States. So access to cheap and high calorie fast food is one of the driving factors for the American obesity epidemic that we observe. And since food is our largest environmental exposure, we consume kilogram quantities of food every day, and those come in contact with our gut, and in all of us, each one of us, harbor trillions of microbes in our gut. It makes sense to take a closer look at the role of microbiome in human health. And if you consider these trillions of microbes in the gut, they make up more than 90% of the DNA in all of us. So we're less than 10% human DNA. And it's not just the sheer amount of DNA, it's the diversity of genes that these microbes harbor. The gut microbiome promotes our immunity and serves as a barrier against pathogens, and it also helps us metabolize the food and get energy that we need out of the food, and also helps with synthesis of vitamins and amino acids. So, as I said, it makes sense to take a closer look at the role of microbiota in human health. And one of the best examples that serves to demonstrate the role of microbiome in human health is a study that was performed, I think now 12 years ago, by Stan Hazen at Cleveland Clinic and his team. And this is from a paper that Zane Wing and his team published in Nature back in 2011, where they looked at a group of people that within a 3-year developed some a major cardiovascular event or stroke or death. And they took these 50 cases and age and gender matched them to 50 controls and looked at to see if there are any patterns that would track with predicting the incident risk of cardiovascular events. And they performed untargeted metabolomics, LCMS in particular, and they also had a validation cohort, a small validation cohort, 25 cases versus 25 controlled. And again, looked at all the analytes that would be highly associated with predicting the risk for these adverse events. And they landed on 18 analytes that between the two cohorts were highly associated with increased risk of adverse events in these individuals. And they were quite reproducible in terms of predicting the risk. And when they looked at these analytes, they found that there are 3 metabolites that fall within the phosphatidylcholine metabolism pathway that track really nicely with predicting these events and also a risk for cardiovascular disease. And these metabolites are choline, betaine, which is not shown in this plot, but it's an oxidation product of choline, trimethylamine, and trimethylamine oxide, or TMAO. And what they observed was that when we ingest food that's high in trimethylamine containing compounds such as phosphatidylcholine, the gut microbiome can cleave the trimethylamine moiety off, and in the liver and all of us, we have enzymes, flavin containing monooxygenases that then convert TMA to TMAO. And up until this publication, the TMAO was considered to be a waste product of this metabolic pathway. But through the work of Stan Hazen and his team, it has been shown over the years that it actually plays a major role in cardiovascular disease and atherosclerosis. So if we, they also went ahead and looked at challenges, choline challenges, and labeled phosphatidylcholine challenges in mice to see if microbiota was truly involved in production of TMAO. So in this case, in mice, they had fed the mice labeled phosphatidylcholine, and they measured the concentration of the 3 metabolites that they had discovered: TMAO, choline, and betaine. And you can see that there is elevated levels of all 3 when there is intact microbiota. After an antibiotic challenge where you wipe out the microbiota, the TMAO levels go down to nearly 0. And after the mice are conventionalized, so they're put in cages with mice with intact microbiota, the TMAO level comes back. And they also looked at the aortic lesions in these mice and saw that the concentration of TMAO in plasma tracked pretty nicely with the lesion area in these mice. So then the question was, are vegetarians and vegans protected? And in a second study that Dr. Hazen's team conducted, and this was published in 2019, they also uncovered an intermediate in this pathway that previously was unknown. This particular intermediate is in between the carnitine and the TMA, and they wanted to see if they give a carnitine challenge to the omnivores and the vegans and vegetarians, if they would have the same capacity of converting carnitine into TMAO, and therefore would be at the same level of risk for cardiovascular disease. So what they observed when they gave label carnitine challenge to vegans and omnivores at baseline, you see that the vegans and vegetarians have almost no capacity in converting the carnitine to TMAO. So the plasma TMAO level is pretty low in vegans and vegetarians compared to the omnivores. But that's not the case with the gamma butyryl betaine, which is that intermediate before conversion to TMA. And in that case, actually, if anything, vegans and vegetarians have a slightly higher plasma concentration of that intermediate. Then they wanted to take a look at to see if at baseline and after antibiotic challenge, whether we could see a difference in omnivores in terms of producing the intermediates. And there was no significant difference in the concentration of this intermediate in the plasma of omnivores. If you compare again the vegans and omnivores, in terms of this particular intermediate, there is no difference in the plasma concentration. But what does that mean for the other analytes? And that's what they did. They looked at the feces of these subjects, because then the conversion must happen. They must be getting the choline through the small intestine into the colon, for example, a distal site. And that's where maybe the conversion is happening and the chemistry is happening. And so that's why they looked at the fecal samples from these subjects, and they saw that the difference between the concentration of the intermediate and the TMA was pretty significant in all diets. And you can see that both vegans and omnivores can display this higher concentration of the intermediate, but not compared to the TMA. And the vegans, however, the omnivores, however, have a higher capacity to convert the labeled carnitine to TMA and the intermediate into the TMA, but not the intermediate step in between. That is insignificant. So then what they did was they wanted to look at another study in humans where they had a cohort of omnivores and vegetarians at baseline, and they gave them the carnitine challenge, and they saw that at baseline for vegetarians, there is no capacity in converting the carnitine into TMA, but in omnivores, you get a spike immediately followed by a lag time. And these individuals came back for 2 visits at 1 month, and then after 2 to 3 months. And you can see that in terms of omnivores, the difference between the first and second visit is not that much. It's insignificant, but the vegetarians and vegans continue to make more of the TMA, which means that there is some reprogramming or shifting in the microbiome composition in vegans after this chronic diet, after this chronic carnitine challenge. So then after these publications came out, and working at the Metabolomics core of Cleveland Clinic with Dr. Mark Brown, we wanted to look at a human relevant diet. So traditionally, mouse models that are fed high fat diet the diet does not resemble anything that humans consume. So in this study, we wanted to give mice a single meal equivalent of fast food, something that a lot of us have consumed or still consume, and see if that challenge would change the gut microbial community enough to produce a physiological change. And so they took, full disclosure, I did not perform the mouse study. There's a trained team of professionals at Cleveland Clinic that performed this study really well. And so they took 6-week-old mice, male mice, and they put them on 1 week of antibiotic. Either they did not, or they did put them on 1 week of antibiotic diet, so with and without antibiotics. And they collected portal blood, peripheral blood, liver, and cecum only after 4 hours of consuming the diet. And the diet consisted of chow, normal chow diet, and what you would get from any of your favorite fast food restaurants. And I'm not going to name these restaurants. You can read them in the method section of the paper. But essentially, we made slurries. The team, the Mark Brown's team, made slurries of these diets, including some of the milkshake and other drinks that people like to get along with their fast food meal. And these slurries were gavaged into the mice, and samples were collected 4 hours later. So this is most papers that you find looking at diet go days or weeks. And this was the first study that looked at such a short amount of time to see if it was enough to produce enough change in the microbial communities that would produce ultimately a physiological change. What I want to highlight here is the importance of collecting portal blood in microbiome studies, which drains the gut, and you can see we wanted to sample that blood because the microbial metabolites are going to be at the highest concentration in that blood compared to the peripheral blood, where the liver has acted upon some of those metabolites and has metabolized them into secondary metabolites. So the 4-hour time frame was decided based on previous studies that's listed here, that TMA and TMA production peak at 4 hours. And so the plasma TMA level peaks at 4 hours, and we wanted to sample that time to where the microbiome activity is at the highest. And so the first thing that we looked at was to see if that single fast food meal after 4 hours was enough to cause any change or shifts in the microbial composition. And this is what you see in this plot, that the chow diet, the normal chow, is separated from the fast food, and also the fast food diet, and also the chow after antibiotics treatment. So what are those changes? Some of those changes you can see in this plot comparing the chow to fast food. You can see enrichment of Enterococcus and Clostridium in the fast food diet, and also loss of Trichobacter and Bacteroides, which are beneficial species in the fast food fed mice. And if you compare chow to chow after antibiotics, or fast food to fast food treated antibiotics, again, you see an enrichment of some of the deleterious bacteria versus the beneficial bacteria. So we went ahead and performed untargeted metabolomics and targeted metabolomics on plasma, both portal blood and peripheral blood, and liver tissue of these mice. The instrument we used was a Q-Exactive HF, and we used reverse phase LC, and the gradient is listed there. We spiked our samples with internal standards so that we could track certain classes of compounds. We used a in-house software that's based on the XCMS algorithm, but does more than what XCMS does, and that it takes into account in-source fragments when it tries to putatively ID the features. And then we fed the features into another open source software called Metabolizer, and it has a suite of statistical analysis that you can generate all of your favorite plots, PCA, volcano plots, and also do some putative IDs against HMDB, KEG, biopsy, lipid maps. And Compound Discover was used for identifying or chemical identification of the features based on their MSMS that we acquired during the run. And the library that Compound Discover uses is MD Cloud, which was sufficient in this case in terms of identifying the compounds. So just to go over quickly what Metabolizer does for post-processing, as I said, you can get the typical PCA and volcano plots that you see in most Metabolomics papers. It's open source, you can download it and use it if you want. It does data transformation normalization and statistical analysis with multiple hypothesis testing, and it does have several small molecule databases for compound ID. And this is just what I just explained. If your data needs any normalization, you can perform Gaussian normalization in Metabolizer. We usually log transform our data, which is what you're going to see in the data that I present. And it's a useful software in terms of unique metabolomics data analysis. So what we had to do, because we had several variables in a study, we broke it up into portal blood, peripheral blood, no antibiotics, chow diet, fast food diet. And so we had 8 different comparisons that we carried out. In this case, you're only looking at this particular comparison, so normal chow versus fast food. In portal blood, in mice that did not get the antibiotics challenge. And if you look at this data, the groups are well separated. And if you look at the metabolites that drive this separation, they fall within the biosynthesis of unsaturated fatty acid metabolism, arachidonic acid metabolism, linolenic acid, and lysine degradation. And those are all important pathways in cardiovascular diseases. So what we wanted to look at was set certain criteria to look at this data, to break it up so that we're only looking at putative metabolites that have a both diet and microbiota dependent response. So out of the 3,800 total ions, we wanted to look at those that were higher in the portal blood compared to the peripheral blood. We also wanted those ions to be higher in abundance in the control groups versus the antibiotics group. We wanted those metabolites or putative metabolites to be higher after the fast food diet compared to the chow diet. So once we did that and put all those criteria on the data, we ended up with 463 ions that met all of these criteria. And out of those, we ranked them based on their fold changes, and these 3 were in the top highest fold change observed. One of them we validated and ID'd as LPA 18, but 2 of them, one had a putative ID and we could not really validate it, and the other one had no ID based on current databases. And you can see that in these plots, what you're seeing is that there is a response where the concentration or abundance of the putative metabolite is higher in fast food diet compared to the control diet. It goes, that response goes away or is significantly decreased after the antibiotic challenge. And it's higher in the portal blood compared to the peripheral blood. So what does that mean for the hepatic metabolism? Because that's where a lot of the metabolism for these secondary metabolites happen. And we know that there are studies that suggest a diet and both microbiota dependent gene expression of in liver. And that's exactly what we looked at, the gene expression or transcriptomics of the liver. And we saw that with respect to chow and fast food diets, you do have an upregulation of a lot of these genes, but that is not the case after the antibiotic treatment, which means that the presence of microbiota is required to see these changes. Same goes for the metabolites. The Metabolomics, this was used by with a Biocrates kit, P400, and the metabolites that you see on the left-hand side showed the greatest difference in the top 50, actually, of the most perturbed metabolites after the fast food diet. And the significance goes away in after the antibiotic challenge. Some of the metabolites that I guess would warrant a second look are listed here. Carnitine or 2-acetylcarnitines that show a microbiota independent change in their plasma concentration, and there are two metabolites here, phosphatidylcholine 32-1 and acylcarnitine 2, that only showed a difference with respect to control versus antibiotic treated. So that's only a microbiota dependence here and not a diet dependence. And just like the untargeted metabolomics study, we also wanted to look at metabolites that showed both diet and microbiota dependencies, and spermidine and H1, which is a monosaccharide, showed this type of response where there's both the diet dependence and microbiota dependency. So the second half of the project was to see if after just 4 hours of fast food diet and seeing all these changes in the metabolome, if that would be enough, if the changes in the gut microbiome communities and the metabolome would be enough to produce a physiological change. Excuse me. And we had seen in papers that host circadian rhythm or disruption of host circadian rhythm is associated with cardiovascular disease, dyslipidemia, and other chronic conditions. And so we wanted to see if we could also see this, because there was a study that came out that showed that the gut microbial dysbiosis could also shift the circadian clock in the host. And so we wanted to see if that was the case in our study just 4 hours after feeding the mice fast food diets. And this is what you see in this group in this transcriptomic analysis of the liver tissue. And what you see here is that the chow and fast food fed mice are completely separated in terms of their transcriptome against the ones that were fed antibiotics. And if you look at this panel of the transcriptomic panel, you see that actually FMO3, somewhere down here, you can see, which is responsible for converting TMA to TMAO, is one of the genes that showed change with respect to fast food diet, and it's actually higher in the fast food fed mice than chow, and that's because of the amount of red meat that is in the fast food compared to the chow. And the pathway analysis of this data also showed that circadian rhythm disruption was one of the pathways that was hit, and RT-qPCR was performed. And in fact, there are two genes where the mRNA expression of these two that are shown down here were shifted after the fast food diet, and these both are involved in regulation of the core circadian clock and the host. And so in terms of inducing a physiological change, that's on transcriptomics level, that's what we ended up seeing. There's a lot of additional experiments that can be carried out after this study. Obviously, the compound ID for the metabolites that we could not identify, and there were a lot of them, is one of them. And also in this study, I hope you realize the importance of portal blood versus peripheral blood for microbiome-focused metabolomic studies, where you have the highest concentration of microbial metabolites. And also acute versus chronic feeding, and what effect can that have on circadian rhythm. And as you saw with the previous Stanhazen studies with the vegans, long-time vegans, more than 1 year vegan, you do have the ability to change the microbiota in a way that it would produce TMAO and at a higher level than a normal vegan. So because the microbiota is plastic, and chronic feeding can induce those changes. And with that, I would like to, went over this already, so I'd like to thank the teams at Cleveland Clinic, the Mark Brown's lab, Luke Osborne, and Daniel Robbie, Amanda Brown, and the rest of the Brown lab members who carried out a lot of the non-metabolomic analysis of this project. And the department chair, Sam Hazen, the Metabolomics Core Director, Belinda Willard, the Microbial Sequencing Core Director, Mr. Sangon, and at National Institute of Standards and Technology, Thijs Mack, who wrote Metabolizer, and the new software that we use for pre-processing metabolomics data. And the funding agencies are listed on here as well. And with that, I will take any questions.
All right, thank you so much. So, first of all, thank you so much, Dr. Goudarzi. That was fantastic, and it's frightening at the same time. You know, we gotta think about our habits, I guess. For now, let's first start with the room, if there's questions. And since this laptop is also the laptop of the host, I will now see if there's also things in the chat room. Right, so are there questions in the room already? Yes, please, Sarah first.
In that first slide, you showed the microbiome. Why was there such a spike in microbiome in the biopsy?
It's not so much a spike, it's the, you're talking about the 16S RNA sequencing.
Yeah.
So it's not so much a spike, it's just the differences in the different bacteria genera that you see. So it's just they're colored differently, but that doesn't mean there is a spike. It's just that they're shifting either from, you know, more Bacteroides to less Bacteroides, or, you know, more Trichobacter to less Trichobacter. So it's not a spike, although it might have seemed that way.
And the other question I had is, clearly it shows that the portal of peripheral blood is important. I know you said you didn't do the mouse stuff, but do you know how that blood is collected differently?
I'm pretty sure it's in the paper, but no, I don't know the details of the mouse, the experiment that were performed on the mice.
Jeremiah?
Yeah, you mentioned the term dysbiosis, and I looked that up, and the definitions are kind of vague.
Yeah.
If you could explain what you mean by dysbiosis, and also, is there a way that doctors diagnose that in the clinic?
So what I mean by dysbiosis, and whether there is a way for the clinicians to measure it,
Yep.
Correct?
Yes.
So dysbiosis is any observed shift from a healthy donor microbial community to anything else. So that shift is called dysbiosis, essentially. is there a way for clinicians in the clinic to measure it? They would have to order 16S RNA sequencing, and I'm not sure if insurance companies cover that. Yeah, so that's the only
It's too
thing.
new.
It's not new, but it's not, I'm not sure. I don't think it's a clinical it's used as a clinical diagnostic tool.
So we now have also some questions online. And if there are other people who are online and have questions, please post them into the chat room. And I think it starts here.
Is there any mention of whether there was more vegetarians than vegans in that group of carnitine challenge? That's a good question, and I don't know the answer. I would have to look that up, and actually, I would have to ask that, because that study was carried out in Dr. Hazen's lab, so I would have to get the details from that team. Are there time points after the fast food or studies that show the metabolome and gene expression tune back? We didn't do that experiment. We did not. After 4 hours, the mice were sacrificed, and we don't have a way of knowing whether they would, if the metabolome or the gene expression would go back after the challenge. I have a general question regarding metabolomics in clinical studies. Do you prefer dependent or independent data acquisition for loaded question for MS data? So if you have software that can handle data independent acquisition, and if you have an instrument that you have verified workflows and methods, sure, but most of what I showed DDA data, and that's because the level of certainty you have with respect to what the signals you're actually seeing are. I personally do a lot of DDA analysis and not much DIA in metabolomics.
Jeremiah has another one here in the room.
Yeah, so I remember you had a slide of your overall workflow, and you showed your XCMS in-house derivative as a separate part of the workflow versus Compound Discover. I haven't used either of those software platforms, but I've heard a little bit about them, and I kind of thought that they do sort of the same thing. Can you explain how they're different?
So the vendor specific software, we don't know much about the behind the scenes. As I understand it, it is XCMS, so it's scent wave based peak picking algorithm. What the reason we used Compound Discover, and what we used Compound Discover for, was compound ID, not so much for the statistical analysis.
But doesn't XCMS also do compound ID?
No, it doesn't. It's just peak picking. That's it.
So, okay, we have a little congratulation here in the chat room. You know, there was, of course, many questions after, but what I was also interested in myself is how big is the difference in these different types of blood that you collected, the portal blood versus the hepatic blood, and how do you collect it?
I can't tell you how we collected the portal blood. I don't know how. I have to reach out to the team who actually performed that part of the experiment. And there are differences when you look at the microbiota enriched metabolites. You would see higher concentration of those in the portal blood versus the peripheral blood. So if you're looking at the peripheral blood, you get pretty much a combination of both host and microbiota metabolism, versus in the portal blood, you get a high concentration of microbiota.
And yes, I understand that. And how big is the difference? Like, is it tenfold, or is it out of, let's say, 3,000 features, you had 50 compounds that were different? Just give us a flavor of what are we talking about here in terms of like magnitude?
It's larger than tenfold. So if you're looking at the microbial metabolites, it's larger than tenfold in the portal blood versus peripheral blood, for sure.
Okay, that's good to know. I'm now curious how to get this. Is there anyone around in the here who would know that? Any one of the participants? Yes, Sarah knows. Okay.
I just Googled it, and I guess the peripheral blood is drawn from the venous sinus by the eyes, and then the
So
these mice were sacrificed first. That's why I say I have to confirm with the team exactly how the blood was collected.
yeah. So, and when, was it alive or was it dead?
Those details I have to ask, but it was done at 4 hours when they sacrificed the mice.
Okay, very good. So since you have also great experience as a core leader before, and then you have now joining a new role as senior person at a contract research laboratory, what is your take on core services versus collaborations? At which time, at which point, what is your advice giving people who are doing work in metabolomics on that interface, talking to biologists, but also performing good metabolomic sciences? How do we deal with that? That's a more generic one, but it's interesting, I know, for people here in the room, but also I'm sure on the web, because that is always this balance, right?
Right. So core services versus collaboration model. Each one is useful if you know what the end goal is and what you want to get out of it, and if you don't abuse that relationship. So with core services, as long as it's not a menu type service, here pick from this menu, as long as there is a more conversation upfront, and each project is really approached individually upfront to see what the needs of the PI is and what the needs of the study is. So when you deliver the data, it matches up to the objectives of the study. But in terms of the collaboration model, I personally think that there is a ceiling for that, and there's a capacity that a lot of labs may not have for the collaboration model. So depends on the type of facility.
All right. What are the 3 most important points that you see as a MANA board member to be important for the metabolomics community to solve in the next 10 years? So what is what we as a community should address 1, 2, and 3?
So standardization in the field of metabolomics is a big issue. And even if in one lab you have, you perform a study in one lab and you come back 2-3 years later, staff has gone through a turnover, your data may not match up against what you have from the previous work that you did in that same lab. The cross institution, I don't even know how to address that, because the challenge there is so large at the moment that reproducibility in metabolomics data across institutions is almost non-existent.
Ooh, So is it a science then?
Okay. So that's the biggest challenge I see standardization in the field of metabolomics. Two is software development, which also is a big challenge in metabolomics in terms of, you know, labs usually have their own workflow, and they say, oh, it's an in-house tool, which, as you saw in my presentation as well, we have an in-house tool. But then if that's the case, how do we standardize the field? So those are the top two challenges I see with the field.
Thank you. Okay, there is a comment in the room.
I had one quick question, if I could, and I don't mean to grill you as the speaker. Do you think that the menu approach might actually help with standardization?
And repeat.
Does the menu approach help with the standardization? It could. Again, it depends on which lab you're talking to and what facility you're recruiting to do your work. Certain fee-for-service entities do not share enough of their methodology. So standardization cannot happen if you don't know how the data is generated.
All right. So with these final comments on the huge challenges ahead, we look back at a very interesting presentation. And again, that makes us double think about who we are and who we are not and who we want to be in terms of, well, metabolism and diets, and the impact on cardiovascular events that you talked about, which is, of course, something that is a leading cause of death in the United States and elsewhere. With that, thank you so much, Dr. Goudarzi. Thanks, everyone, and goodbye, and see you soon.